everyone, myself I'm student of 11th standard from Nyanadi Institute. Today I am going to explain ETS from chapter number 12, Respiration in Plants, under the guidance of Mr. Suleiman. So firstly, where does ETS takes place? So ETS takes place in the inner mitochondrial membrane in crystal. After that, what is mean by ETS? So ETS is a metabolic pathway where electrons are carried from one carrier to another carrier. The overall purpose of ETS is to make ATP by using NADH and FADH. After that, what is the real mode of ETS is to increase the number of protons in the inner membranous space comparatively to the matrix. So now we will have greater number of protons in the inner membranous space than in the matrix. After that, we know that there are 5 complexes in ETS. Firstly, complex number 1. It is known as NADH dehydrogenase. There are chemicals like FES and FMN. Now, why NADH dehydrogenase? Because here dehydrogenation of NADH occurs. What happens? Dehydrogenation of NADH occurs and we form NAD. And this, after dehydrogenation, 2 electrons. By taking 2 protons from the outer side, we will obtain here 4 hydrogen atoms. Nextly, the electrons of FES will be given to ubiquinone, and this ubiquinone will use the electrons and make the reduced ubiquinone, which is also known as ubiquinone. This is also known as ubiquinone, which is made by reducing the ubiquinone. After that, what happens? There is a complex number 3, which is known as cytochrome BC1. Here chemicals like cytochrome C, FES and cytochrome B are formed. And nextly we have here ubiquinone. Now this ubiquinone and this ubiquinone gets transferred here and we get more 4H hydrogen atoms in the inner membranous space. After that, we will come to the cytochrome C. Cytochrome C plays a main role of carrying electrons to the above complexes. So firstly, cytochrome C is located in the outer surface of inner membrane, and cytochrome C is a mobile carrier between complex number 3 and complex number 4. So it will be here cytochrome C. After that, here we have ubiquinone. Next thing is complex number 2, known as succinate dehydrogenase. Here there are formation of succinate and fumarate. Okay, and chemicals like FES and FAD. In this complex do not accept any number of protons but accepts only electrons. After that, there is complex number 4, which is known as cytochrome C oxidase. The 4th complex is known as cytochrome C oxidase. Now, this carrier, as I said, it is a carrier between complex number 3 and complex number 4. So, cytochrome C will provide protons to the copper A, and copper A will provide it to cytochrome A, cytochrome A3 and CuB. Now during this process there will be reduction of two electrons, and we will get a reaction which is half plus 2H plus, which will give us H2O. What is the work of O2? O2 takes hydrogen and converts it into water molecule. After that, we will go for the after that what is the function of ubiquinone. So ubiquinone takes hydrogen atoms, hydrogen electrons we can say, from the matrix and transfer it to this side in the intermembranous space. After that, by the reduction of 2H in the intermembranous space, we will have only 2 hydrogen atoms. After that, there is fifth complex where there is synthesis of ATP. ATP synthase. Now, these 2 hydrogen atoms from the inner membranous space will travel to here by having facilitated diffusion and ATP synthase, and then we will get H plus atoms on the outer side. And after that, there will be ATP synthesis and we will form the products like ADP plus P dash and ATP. And production of ATP is only known as oxidative phosphorus